Hi, Science 10. Today our lesson will be 8.1 on the parts of the cell membrane and 8.3 cell size. They're both really little topics, so we'll talk about them together in this one video lesson. The cell membrane is this outer layer that is on the outside of an animal cell, and plants also have a cell membrane that's underneath their cell wall. That cell membrane protects it from the outside environment, and it also has other functions like allowing substances into the cell. Some substances are not allowed into the cell. That membrane will also fold in and become a membrane around vacuoles that are pulled in, and will also, if they're expelling vacuoles of materials or cell products, it can fold into that membrane and be expelled that way through that phospholipid membrane. The membrane that is found around the cell has the same components as the membrane around all the organelles. So around lysosomes, ribosomes, Golgi, ER, all the membranes of those organelles are also the same structure. And the structure is that there's a phospholipid bilayer, and these phospholipids look like this. You can see that the tails are all facing in. That's because it's watery on the outside of the cell and watery on the inside of the cell, as the cytoplasm is. And these tails, they call them hydrophobic, so they're scared of water. So those tails will face inwards. The head part of the phospholipid is hydrophilic and likes water. So those heads are near the water on the outside of the cell and near the water on the inside of the cell. You can see in this diagram that you have from your textbook, you've got to imagine that this entire phospholipid bilayer because there's two layers here, surrounds the entire cell. They're just showing you a little chunk of it here, but you can see those heads and tails right here, and that's your phospholipid bilayer. There's two layers of it, and you can see that embedded in that are these really, as you can see here, the red colored proteins embedded in the bilayer. Those proteins have different functions. Some of those proteins are channel proteins like this one, and they have a channel that allows certain materials through. Some of those proteins are cell recognition proteins, so they have some kind of a glucose chain on them, which is individual. They call them cell recognition because every individual has different proteins on their proteins in their cell membranes. So because there's different glucoses on their proteins, your cells of your body recognize cells that belong, and they recognize cells that don't. So other people's cells have different cell recognition proteins, which is what makes organ transplant difficult, because your body will recognize that someone else's organ has different markers or recognition proteins, markers on their cell recognition proteins that are embedded in their cell membrane. So we call this a fluid mosaic model. Some of the video clips that you can watch in this topic will show you how floppy and fluid the cell membrane is. It says that the components aren't attached to each other, so the membrane is very flexible, and if you can imagine something made of a lipid, how soft and sort of mushy that texture of a lipid is. You can have a look at the video to see it in animated form. So the carbohydrates that are found as little additions, it says identify cells that belong in your body. So when we looked at these chains here that are found, sometimes they're even found on a lipid, sometimes they're found on a protein. They will help identify the cells of your body. The proteins are basically structural support and channels for materials to move through, and they would be called channel proteins if they allow substances through. And I guess here's a picture of a channel protein here. Okay, they have done this process of freeze fracturing cell membranes, and it's really neat to see what happens if you pull apart the phospholipid bilayer like they're indicating here. If you pull it apart, what you can see is all of those little bumps left behind, showing you how many and how numerous those proteins are that are embedded in that phospholipid bilayer. So it's really neat to see all these little bumps and see how the freeze fracturing method has exposed all those embedded proteins like you see in these little red proteins in this picture. That's kind of how they do that. Jobs of the membrane, some of them are filters, so they're semi-permeable. Semi-permeable means that certain things are allowed to pass through the cell membrane, others are not. And sometimes it depends on the size, shape, or electric charge of the substance, whether or not the cell allows it in. We're going to flip over to the next topic on topic 3, 8.3, next. And I told you it was really short to do 8.1. 8.3 talks about cell sizes and how most organisms out there have many cells in them, and like your body that's made of 100 trillion cells, that's many cells, and they're very tiny cells. To have multiple cells gives you some advantages. That means that if the cells are very numerous, they'll be very tiny, giving high surface area, and that means there's a lot of, and like your body, 100 trillion cells gives a lot of area for nutrients to be exchanged, wastes to be expelled, gases to be exchanged. Because there's so many cells, there's a lot of surface area for that to get done. Also, with a high number of cells, that means that the cells can become specialized into tissues to do certain jobs. So certain tissues in your body have certain specific jobs that they do. There was a couple questions in your notes that says, how long would it take oxygen to get into a cell that's about your size of your body if you were one big cell? And it would take like a week for oxygen to get into all of the components of that cell, which shows you that that would not be efficient to be made of one giant cell. And that cell in that time would likely die because all of the parts would not be getting oxygen. In the dinosaur era, insects were very big because of the high concentration of oxygen there was in the atmosphere at that time. Okay, next, intercellular transport. It talks about how molecules will get into the cell and start diffusing to where they are needed. And when you have a high concentration on the outside of something like nutrients or oxygen, it will continue to diffuse in where there is less. And if they're small cells, they can diffuse in faster. So it says your volume increases faster than surface area. If you were look at a 1 millimeter cube versus a 2 millimeter cube, the surface area to volume, there's 6 to 1 of surface area to volume in the little guy, which would be a higher rate of diffusion than when you're 2 times 2 times 2, 2 millimeter cube, you're only going to have a 3 to 1 surface area to volume ratio. You go to a 4 millimeter cube, you get a 1.5 to 1 surface area to volume ratio. So this is not very efficient at getting oxygen and nutrients into the center of the cell. The demo that is done in class is that I take a red cube with a red dye that will react and turn yellow in the presence of acid. So what I do with these 3 little cubes is put them in a beaker of acid, and we watch how quickly the acid diffuses and turns the cube yellow. And of course, the little guy, it happens so much quicker. It's turning yellow from all sides. This one, I have the stats, I think, on here. The middle guy, the little guy took like 16 minutes to turn all yellow. And it was a 1 by 1 centimeter cube. The 2 by 2 took 47 minutes, and the 3 by 3, our class finished and it was still going. It was an hour and 25 minutes to diffuse entirely that acid into the center of this little cube. So we're seeing how that would not efficiently sustain life if the cells were so big. So what happens is our cells are small, and your body has even other ways to make sure, well, organisms have other ways to make sure that surface area remains high. So they end up having infoldings like the infoldings of the center of your stomach, for example, or the number of root hairs that that plants have, or the fact that there's not only in your small intestine, the inside not only has villi. You could draw your villi inside of your small intestine. This is the inside of your small intestine, but also it's got little microvilli. So inside your intestine, just to make sure there's lots of area to both digest food and then later on in the intestine to absorb food, all the little villi are covered with microvilli. So you can see how that greatly exponentially increases the surface area to allow nutrients to pass in. Multicellular organisms, like a large tree, it is definitely not one big cell. It doesn't have bigger cells. It just has more cells. So the bigger the organism, the more cells it has. Cells cannot be basically any much bigger than maybe 1 millimeter in diameter would be the biggest cell there is, which is an egg cell of humans. Otherwise, they're all very, very, very tiny, and that makes them efficient at transporting materials. So you can watch this video if you like, and that